discuss capillaries. Keep in mind that the capillary only is made of that endothelium or the tunica interna or the tunica media. It oftentimes will have some slight definition around it. In this case, what is pictured here is a basement membrane, which is typically the type of structure that will be around there that just assists the cells in support, kind of keeping them anchored to the surrounding material and keeping it supported. Okay, so there are three different types of capillaries which are shown here. The continuous capillaries are the most common. These are found typically in the skin and in the muscles, which are large swaths of material in our bodies. In this case, the endothelial cells will provide an uninterrupted lining where the cells are joined by tight junctions. In the case of most continuous capillaries, you will have these structures called intercellular clefts. Intercellular clefts are spaces between the cells, and in this case, they are usually just large enough for fluids and small solutes to pass through. However, there is one special area of the body where the continuous capillaries do not even have the intercellular clefts, and that would be the blood-brain barrier in the brain. So the very important blood vessels that bring blood through the brain and do allow for the feeding of the neurons in the brain do not actually have contact with the neurons. Instead, we have these specialized cells called astrocytes that grab the continuous capillaries and ensure that there is no actual transmission of blood into the brain directly. So in that case, there are not even intercellular clefts. There's a different specialized structure that allows things to pass in and out. The next type of structure we're looking at, the next type of capillary, is called a fenestrated capillary. The fenestrated capillary is a structure that is riddled with fenestrations. As you can see, fenestrations are pores. These are oval pores. Usually these are covered by a membrane. The fenestrations are more permeable to fluids and small solutes than are like the intercellular clefts of the continuous capillaries. This would be in places, you'd find fenestrated capillaries in places where you want active and rapid capillary absorption or filtration of structures. So a really good example of this would be in the kidneys, where you have to make sure that the kidneys are filtering blood properly. And so we'll come back to fenestrated capillaries when we talk about the glomeruli of the kidneys. The next type of capillary is called a sinusoid capillary, and as you can see, it's quite Swiss cheesy. There's holes that are quite large here. The sinusoids here, they have highly leaky capillaries. This allows for example, cells to be able to pass quickly in and out of the blood. For example, white blood cells may be able to be passing back and forth, and that may be very important in certain parts of the body. You'll find sinusoid capillaries in the liver, in the bone marrow, where white blood cells are manufactured, in the lymphatic tissues, where white blood cells do a lot of their work, and some of the endocrine organs. As you can see, the lumens tend to be very large and irregular. There's very little in the way of tight junctions between the cells. It's quite a loose connection, and the intercellular clefts tend to be massive. When we look at capillary beds, what capillary beds are, keep in mind what capillaries are. Capillaries are the structures that actually feed the tissues. And they are the only structures that are able to feed the tissues or remove things from the tissues. So they're the only ones that actually communicate directly with the tissues. Arteries don't do it, veins don't do it, only capillaries. And of course, this only happens at the capillary beds, pretty much. It's special. Not all capillaries are actually capable of going back and forth between, of allowing for transmission of structures and substances from the blood into the tissues and back. So what we're looking at here is kind of a cool way of picturing a capillary bed, where we have the arteriole up above, and then what we'll be discussing next, which would be a postcapillary venule here. And what we'll notice that in between these two guys is your capillary bed. On the left-hand side, we have a capillary bed which is readily feeding the structures surrounding it. And here we have, on the right, we have a capillary bed which is not. So capillary beds are interweaving networks of capillaries. It allows for great efficiency of transport of things into the tissues and into the blood, but there is a vulnerability that the blood has in this area, because this is where it's most dangerous. It's most dangerous to be exposing the blood to the tissues because things happen in tissues, like pathogens might be invading, things like that. So there's a vulnerability here as well. There's a lot of details in this picture, but there's two main types of vessels that I want you to pay attention to in a capillary bed. There is the vascular shunt. The vascular shunt brings blood directly from the arteriole to the venule, and it goes straight down the center here. And then around that are true capillaries. The true capillaries, there will be 10 to 100 true capillaries per capillary bed, and this is the actual exchange vessels. I want you to pay attention to one structure here that's actually kind of hard to see in this picture, but see if you can see it. These structures here are called precapillary sphincters. And this is the first time you're coming across a sphincter, but we'll be talking about these a lot in this part of in semester 2 of A&P. A sphincter is a ring of muscle that basically can squeeze a tube shut or relax and allow a tube to open. So what we're looking at on the left-hand side is that the precapillary sphincters are open and relaxed. And on the right-hand side, what you're looking at are precapillary sphincters that are constricted or contracted or closed. And what this does is like on the right-hand side, what we're saying is that blood goes directly through the vascular shunt and does not actually make its way into the true capillaries. So on the right-hand side, for the most part, this capillary bed is not feeding its surrounding tissues. And I want you to think about examples of why that might be useful. I also want you to think about the fact that the three places where blood is controlled, like the flow of blood is controlled. It's not just at the precapillary sphincters. There are two other places as well, and see if you can come up with that. And jot it down in your notes. Do your best guess, and we'll discuss it in class.